Hello everyone. Welcome to Barcode Biotechnology. Welcome to the second session of our bootcamp drug design and drug discovery. Okay. In this session, we're going to discuss about the drug discovery process, the drug discovery pipeline, and the drug repurposing study. I hope that you are having your pen and paper ready and you have to note down some important points and some concepts which will be learned today. Okay, so let's start our lesson number two of the session. Okay, so let's start the session. So first of all, here it is the overview of computer-aided drug design and its relevance in the drug discovery. So what is the relevance of the computer-aided drug design in the drug discovery? So what is CAD? So basically the CAD is nothing but it is some sort of computational method, algorithms and software tools that are used to accelerate the discovery, the design and optimization of the drug candidates. Okay. So it integrates the principles from the chemistry, from the biology, from the mathematics and the computer science to predict how small molecules can interact with the biological target in the molecular level. Okay, so let's understand by process of the example. Let's suppose we take an example of S aureus. Okay, many students have known about S aureus. Kindly comment in the comment section what is the full form of S aureus and what is its category. Okay. So the S. aureus, it is a bacteria, and it have an enzyme that is the dihydrofolate reductase, also known as the DHFR. Okay, this bacterial enzyme it causes many some sort of bacterial infection. Okay, so DHFR helpful in the bacterial infection. Okay, so in the bacteria Streptococcus aureus, the dihydrofolate reductase is present, which helps to survive the bacteria, and that bacteria causes the bacterial infection. So now we have a marketed drug. Let's suppose we have the marketed drug of fluoroquinolones. Okay, in fluoroquinolones, we have the moxifloxacin. Okay, so now the moxifloxacin is inhibiting the bacterial enzyme dihydrofolate reductase. So the dihydrofolate reductase is inhibited by the moxifloxacin. Okay, so this is the pharmacology or the biology portion. Okay, so in this way, this drug is defining or relevant in the field of biology. Okay, next, how it is chemically relevant. See, if we have the 3D structure of the dihydrofolate reductase, so that 3D structure we can find out from the RCSB-PDB, that is the protein database. Okay, so this dihydrofolate reductase is nothing but it is your protein or enzyme. Okay, so now that enzyme must be prepared of amino acids. So the amino acids have bond like this, that is one carboxylic acid and NH2. Okay, they are also called as the amino acids are also called as the zwitter ions. Okay, so now if my drug moxifloxacin, okay, let's suppose this is one portion of the moxifloxacin. Let me draw like this, and let's suppose here I give the CS3 and here I give the CS3. Okay, let's suppose this is one portion of the moxifloxacin. Now, this OH group can form an interaction between the carbon and this hydrogen OH group. They may form any some sort of interaction. So there are various interactions are there, just like van der Waal interaction. Okay, van der Waal interaction is there. Hydrogen bonding is there. Okay, so in this way, the moxifloxacin, the drug, and the dihydrofolate reductase, the bacterial enzyme are chemically relevant. chemically relevant. Then if we go deeper, after the docking result is generated, we go for some sort of studies that is called as the molecular dynamic studies. In this study, we find out the RMSD, that is the root mean square deviation, the RMSF, the radius of gyration. So basically, all these things are the quantum mechanics things. Okay, quantum mechanics. All this follows the quantum mechanics. So in this way, this interaction also comes the mathematical, and in the back end, as you all know, that all the softwares for the docking that runs in Python. Many of the softwares run in Python language. Okay, so Python, also the computational computer science or the information science, sorry, that runs in the back end. Okay, so in this way, the principles for chemistry, biology, mathematics, and the computer science are also responsible for the interaction of the different small molecules with the biological target. I hope all of you have understand how the chemistry, biology, mathematics, all these things are relevant to the drug discovery. Okay, so going to the next slide. So, what is the main core objective of the computer-aided drug design? So first of all, to identify the potential drug candidate. Okay. To identify the potential drug candidate, as I have mentioned, for the SRS, we have fluoroquinolones. You can Google what is fluoroquinolones. So basically, these are drugs, basically mostly focus on the antibacterial agents. Okay. That is, it inhibits the bacteria like drug moxifloxacin. As I have mentioned, moxifloxacin is there. Ofloxacin is there. Ofloxacin, it is given for the treatment of the motion sickness. And also it is given for the loose motion. Okay. So apart from that, sulfonamides are there. Sulfonamides are there. There is also macrolides are there. So these are the class of antibiotics. Okay. So to identify the potential drug and targets and the drug compounds. Then to design or select the compounds with high binding affinity. Okay, so by using the computational techniques, we can design or some sort of compounds that have high binding affinity towards the receptor. As I have mentioned and given you an example of moxifloxacin. Okay, again, so I have given you the example of moxifloxacin. Okay, so how can we design or any prepare any compound that have more effectiveness as compared to moxifloxacin? We can go for the LBDD. So now what is LBDD? It is nothing but your ligand-based drug design. Ligand-based drug design. Okay. So now what we do in the LBDD, just I am giving the short information. We are going to learn it in this session only. So we have to prepare a set of 1 to 100 compounds of the moxifloxacin. Okay. Means if we have a moxifloxacin, we can prepare the derivatives. Let me show you here only four derivatives. We just have a different substituent in the moxifloxacin. We substitute with OH group. We in moxifloxacin we let substitute with the nitro group. We may substitute with the amino group. We may substitute with the cyano group. So we just remove one position, substitute with another group. So this is called as the LBDD or the ligand-based drug design. So we can design or select compound with high binding affinity. So what we do, we just substitute, then we perform the docking, then we find out the result and we can get the desired compound. Then the third one, to predict the ligand target interaction. So by using the docking process, By using the docking process, we can easily find out the ligand target interaction. We can visualize by using some sort of the tools like BioWare Discovery Studio, then optimize the lead molecule for potency and selectivity. Once we find out more active compound than the marketed one, we go for the optimization. Then we go for the other screening process, which in the next slide we are going to learn about that. Next, the reduced experimental time, cost, and failure rate. So what happen if we perform a or we have to find out a drug by manually? So first of all, we have to synthesize the different derivatives of the moxifloxacin. First of all, the cost of the drug. Okay, cost of the reactant. So performing a reaction, we need some reactant. So who knows the cost of the reactant? For 5 gram of the reactant, it may be 5,000, 10,000 rupees. Okay, so many of the reactant cost higher. Okay, so the next problem that may arise in this section while we experimentally prepare, first is cost is there. Second, if the reaction is going or not, the synthesis will be very much hectic. We cannot prepare a 100 set of compound by just synthesizing, by just purifying that after synthesis we have to purify, then go for the NMR, IR, mass, okay? Different some sort of characterization techniques are there. So we cannot afford that. Okay, the synthesis problem is there, and there is not an guaranteed that we can synthesize that derivatives in the first attempt. We may have failure. So instead of doing all those things, we just virtually prepare the drug molecules. That 2D structure we generate, then we convert it into the 3D, then we perform the docking, all the things we have performed. I think this slide is clear to all of you. So whatever your doubts, you just put in the comment section or you just write there and I will surely answer all those questions. Okay? Okay. So now comes the drug discovery pipeline. Okay, so the very interesting portion, and we have also learned in the previous session also. I am just going superficial. As I have mentioned about that antibacterial drug, moxifloxacin. So we have 1,000 set of compounds we have prepared virtually by using some sort of tools that are mainly used for the drug design, like ChemSketch is there. I am talking about some sort of free softwares like ChemSketch is there, ChemDraw is there, Marvin scale is there. Okay, but in the large scale, there are very other tools are there for the drug discovery. So we have generated an idea. We have to prepare one molecule or one drug compound that is more effective than the moxifloxacin. So we draw the compounds. Then what happens? After that, we go for the molecular docking. We go for the molecular docking. Okay. So after performing the molecular docking, we get various numbers of results. For the 1,000 compound, we get 1,000 of result. Let me give you a short example. We prepare 5 compound, 5 derivatives. Okay. So we prepare the 5 compound derivatives. After that, we go for the molecular docking. For the preparing of these compounds, we use ChemDraw. We optimize the ligand molecule and we perform the molecular docking with using the chimera. After the molecular docking, we get 5 different results. For the L1, we get minus 9.5. For L2, we get minus 7.5. For L3, we get 9 point, again 9.5. For L4, we find the result of 8.7. And for the L5, we get the result of 8.0, all in minus. Okay. More the negative energy, more is the affinity towards the receptor. Okay. So when we perform the same docking using the moxifloxacin as a standard, or reference, we get the result of minus 10.2. So now you can see that the L1 and L3, these two drugs is having a very fair or equal amount of binding affinity towards the receptor as compared to the reference one, that is the moxifloxacin. Understand carefully, listen carefully all these things because it is very much important for you. Okay. So after that, we from these five compounds, we select the two compounds for the further screening. Okay, further screening, that is the L1 and the L3. Okay, so now we have to analyze the ADME property and the another property, that is the Lipinski rule. In the previous session, I have told you about the Lipinski rule. You can go and watch the Lipinski rule of phi. So what the Lipinski rule of phi states that the molecular weight should be less than 500 or equal to 500. Then the topological polar surface area is there. Then number of acceptors should less than 10. The number of hydrogen bond donors should be less than 5. And the log P, that is the lipophilicity value, should be less than 5. Okay. So if the drug, the L1, L3 follows all these five rules, then we go for the further study. We go for the basic process that involves the ADME study, virtual ADME study, toxicity study also, Lipinski rule. Then we go for the synthesis. Okay, we go for the synthesis of these two compounds. Okay, synthesis of L1 and L3. Okay, so after the synthesis, we found out that L1 and L3 has been synthesized. Then we go for the clinical trial. In the clinical trial, we go for the phase 1 study. In the phase 1 study, we perform the apply the in vitro analysis towards the animals. Okay, animals like rat, like mouse, then guinea pig is there. In this phase, we studied an animal and we see the better tolerance of the drug molecule and suppression of the receptor, that is the S aureus receptor dihydrofolate reductase is taking place. Then we go for the phase 2 study in which we apply this drug compound to the human bodies, okay, in a small scale. In the phase 3, we apply this drug in a large scale, more than 1,000 people. After confirmation of all these things or passing the phase 1, phase 2, and phase 3 of the clinical trial, then we apply for the regulatory approval. For the applying of the regulatory approval, we have to submit a form that to the FDA, that is the Food and Drug Administration. Okay, that is the full from is US FDA, that is United States Food and Drug Administration. After getting approval from the US FDA, the drug is be marketed for the patient care. Okay, so after getting that, we go for the phase 4 analysis. So in phase 4, what we do, we evaluate the long-term side effects or the long-term adverse effect of the drug compounds. Okay, so this is the phase 4. Okay, so now next, what we have to do, we have to go for the types of drug design. Okay, so basically there are two types of drug design are there. Two types. The first one is your structure-based drug design and the ligand-based drug design. Now here comes a tweak. The structure and the ligand. What is the difference? You can just think that the structure means it's basically the structure of the drug molecule. But no, here the structure, what it means, the structure, it is the 3D structure of the biological target. By taking the same example of dihydrofolate reductase, so we have to find out what is the 3D structure of our target. Here the 3D structure is our dihydrofolate reductase. It may be enzyme, the structure of target may be enzyme, it may be receptor, it may be protein, or it may be the DNA. Okay, so DNA is also one some sort of target. So what are the techniques that includes in the structure-based drug design? That the molecular docking is there, binding site analysis. So the binding site analysis is done by using a tool that is called as the CASP-P. Okay, by using the CASP-P tool, we can find out what is the binding pocket. In the previous session, I have told you about the binding pocket. So what is the binding pocket? Let me show you. So in this receptor, you can see that the drug molecule is present inside the receptor. So this position where the drug molecule is placed, this is called as your binding pocket. Okay. Next, what are the other techniques? That is the molecular dynamic simulation, which I have told. That is the RMSD and RMSF values. So these are the simulations. It is commonly applied when the target crystal structure are available from the protein data bank. If in the protein data bank our crystal structure is available, we can go for the structure-based drug design. Okay. Now next come the ligand-based drug design. So the ligand-based drug design is used when the target structure is unknown. Okay. If our target structure is unknown, we can go for the ligand-based drug design. It mostly relies on the known active compounds. So, what are the techniques in the ligand-based drug design? That is the pharmacophore modeling and the QSAR. So, now let's take an example of the pyrimidine ring. So, here you can see that is a pyrimidine. It is a heterocyclic moiety. It is one of the base, that is the pyrimidine base of the DNA. So, we have two types of base. One is a purine base, another is a pyrimidine base. Okay, so we have two types of base, purine base and pyrimidine base. So this pyrimidine base, you can say that this pyrimidine base is present, or the pyrimidine moiety is present in the antimicrobial agent. We just substituted at various position of this pyrimidine, and it is acting as an antimicrobial agent. So antioxidant agent, you can see there is this complete entire position, the benzene ring containing three methoxy group, and here is a thio group is substituted. Okay, by just substituting all these things, it acts as an antioxidant agent. It is also used as an anti-malarial agent. Here it is a piperazine moiety is attached. Anti-inflammatory, it is also act as an anti-inflammatory. Also, it is act as an anti-cancer, anti-leishmaniasis, antiviral. All these things you can know that the pyrimidine ring can produce. Why there might be some chance it act as an other other properties it may have that is been underexplored. Okay, many of the scientists are working to find out that if we have a heterocyclic compound, let's suppose we have one structure that is a chromin ring, which is majorly majorly research currently the chromin ring. So benzene ring containing another benzene ring containing oxygen, it is called as the chromin. So the benzene ring containing another benzene and the nitrogen, it is called as the quinoline. Okay, so all we know that the quinoline, it is an anti-malarial. Chloroquine is there, anti-malarial, and as well as it is the anti-inflammatory. It acts as an anti-inflammatory agent. Who knows that by the applying of the QSAR, that is the quantitative structural activity relationship. We just replace here, I add the OH group here, and it can act as different disease. It can use for the any other different some sort of disease like antiviral or antifungal. Okay, so it is getting explored currently, the ligand-based drug design, and in the market and in the current research, mainly focusing on the ligand-based drug design. Okay, because many of the researchers are synthesizing many moieties and they are finding out the target molecule by just going through that ligand molecule. Okay, next. the drug repurposing studies. So, what is drug repurposing studies? So, before going to drug repurposing studies, let's go through some softwares for the drawing of the ligand molecules and also go through the Swiss ADME where we analyze our drug molecule. Is it following the Lipinski rule? What are the physiochemical parameters of our drug molecule? Okay, so let's go for this. Keep it here. Here, this is the interface of Swiss ADME. You just have to go to Chrome and search for the Swiss ADME and this interface will appear. Okay, so you can upload any some sort of the compounds formula like you can see that there are different formats to upload your compound like MDL format is there, HDF format is there, HD file format is there, then Smiles format is there. Okay, so you can let me show you how can you just put your compound here by using this process. Okay, so for that you have to go to PubChem. So it is opening the PubChem. So in PubChem, what you have to do? We have to search for the molecule. For that, let's suppose moxifloxacin. So PubChem is the database where you can get a number of millions number of compounds database. It is okay. You can find out any some sort of compound here. Okay. So you just have to click on moxifloxacin. So you can see that in the moxifloxacin, all the content, it is given as the content in the right side. You can see content, the structure is given, then what is the primary laboratory hazard? It is irritant in nature. You can see it is written that it is irritant. The molecular formula, the synonyms, then the molecular weight is given, that is the 404.4. Then it is given the description, moxifloxacin, it is the fluoroquinolone class. Okay, then the 2D, that is also given the reference compounds also like other derivatives of the fluoroquinolone. Ciprofloxacin is there, ofloxacin is there, norfloxacin is there. So this is the 2D structure or the 2D representation of your moxifloxacin. Then the 3D conformer, you can see this is the ball stick format of your moxifloxacin. Then you can just go for only stick format also, wire format also. Okay, all the information have given here. Then this is the IUPAC name that is given of the moxifloxacin. INCHL, it is another code name for the moxifloxacin is there. And you can see that is the easiest structural representation, that is smiles is given here. You just need to copy the smile for the Swiss ADM. And if someone is going to order moxifloxacin, let's suppose you are using moxifloxacin as a reactant. So for industrial purpose, there is cash number is there. So basically, this cash number is nothing but if you want to order or purchase any chemical from the industry, you just have to provide the cash number. Okay. Other informations like what are the chemical and physical properties, the spectral information, that is the 1H NMR, carbon 13 NMR, all these things. Okay. And the chemical vendors, toxicity. Just go through this one and you can understand what are the different things that are given in the PubChem. Okay. PubChem is nothing but it is a library for the chemist. Okay. The drug discovery persons and also the synthetic chemist. Okay, so we have to go to the Swiss ADME. We simply paste the smile, then click on add. So based on the smile, it will generate the structure here. It is taking some time. So if you don't want to prepare or don't want to copy paste the smiles and get the structure, using simply you can draw the structure by using this benzene, all these parameters that have been given. Okay. For today's session, we are going to discuss about the moxifloxacin. So the moxifloxacin structure you can see here. Then we click on this one and it will generate the smiles again. Now we can just simply press on the run and it will show the all the parameters of the moxifloxacin. Okay. So as I am talking about the ligand-based drug design, let's suppose we change the OH group into the NH2 group. Okay. So the first one, don't get confused. The first one, this is the moxifloxacin one. And the second one, I have replaced the OH group with the NH2 group to find out what is the difference result showing between the moxifloxacin and the NH2 substituted moxifloxacin. Okay, I press and run, and it is running right now. I hope that all the students are getting all the things, and if you have any doubts, kindly put that doubt in the comment section. I will answer all your doubts. Okay, so you can see the molecule number one. Okay, in the Swiss ADME, the molecule number one, it is the moxifloxacin, and the molecule number two, you can see that it is represented by the NH2. Okay, so what are the difference? Let's see. Okay, the structure is loading, but we can analyze that. First, the physiochemical properties. Okay, the physiochemical property. What are the physiochemical properties? The molecular formula is given C21H24FN3O4. The molecular weight, that is the 401.43. Number of heavy atoms. So what is heavy atoms? Here comes the heavy atom. You can write down that apart from the carbon, all other, okay, apart from the hydrogens, okay, apart from the hydrogens, all other compounds that are present, that is called as the heavy atoms. So you can see there are 29 heavy atoms. Let's just forget this H24. Now calculate C21. Here 21. Fluoro is also an heavy atom, 22. Nitrogen is the heavy atom, 21 plus 1, 22. Nitrogen 3, 23, 24, 25. Plus oxygen is 4, that is 25 plus 4, that is the 29. So number of heavy atom here is 29. So apart from the hydrogen, all the structures or all the compounds that is present is called as the heavy atom. So we have calculated number of carbon 21, fluorine plus 1, that is the 22, nitrogen 3, that is 25, oxygen is 4, 25 plus 4, that is the 29 number of heavy atoms. So what are the number of aromatic heavy atoms? The 10 numbers of aromatic heavy atoms is there. Number of rotatable bond, it is the number of 4 number of rotatable bonds are there. Talking about the hydrogen bond acceptor, that is 6 hydrogen bond acceptor. Following the Lipinski rule, number of hydrogen bond donor is 2. Then the topological polar surface area, it is 83.80 angstrom. The log p value or the lipophilicity is 2.78. It should be less than 5. It is 2.78. That is less than 5. Then the log S, that is the water solubility, that is minus 2.70, and it comes under the class of soluble. Okay. If it is more than 10, less than minus 10, it is insoluble class. For the poorly soluble class, minus 6. For the moderately soluble, minus 4. For the soluble class, that is minus 2. And very soluble, less than 0. And highly soluble is the result come in the plus. Okay, so it comes under the class of the soluble. So where it is soluble? It is soluble in the GI absorption. When the drug undergoes inside our body, it start to dissolve. First it dissolves, then it gets disintegrated, and it flows in the blood. There is one terminology is there, that is bioavailability. That is amount of the drug that reach to the blood circulation. When we take the drug, goes, it get dissolved, then it get disintegrated. After that, it flow to the blood circulation. So the all the 100% of the drug is not going to the blood circulation, systemic circulation. So amount of the drug that goes to the systemic circulation, this called as the bioavailability. I hope that all the students are writing all these things. You can pause the video or go backward and again listen to these things because these things are pharmacology based things and you are needing that things. Okay. Next, pharmacokinetic parameter. It do not cross the blood-brain barrier. Okay. Then paraglycoprotein substrate. Yes, it is having the paraglycoprotein activity. It inhibits. Okay. There are major cytochrome P enzymes are there. Cytochrome P1A2, 2C19, cytochrome P3A4. It doesn't inhibit the enzymatic activity of the cytochrome enzymes. Okay. Next comes the drugability or drug likeliness study. It is not following, not having any violation. It is following the Lipinski rule of five. There are other parameters like ghost parameter is there. According to ghost, the molecular weight should be less than equals to 160 or 48. Okay. So you can go through this ghost parameter, Weber parameter, again Muge bioavailability score that is 0.55 and on the medicinal chemistry point of view, it is synthesis accessibility 3.89. So what is synthesis accessibility? From 1 to 10. The scores that come under 1, it is very easy to synthesize. The score that comes 10, it is very difficult to synthesize. Okay, so it comes under 3.89 approximate. We can say it is 4, and it have moderately difficult to synthesize, easy to moderate. Okay, so now next come to the second drug that is the we have prepared the derivative by using the LBDD. The same thing is there, the physiochemical parameters. The molecular weight is 400, number of heavy atoms 29, number of aromatic heavy atoms 10, rotatable bond is 4, hydrogen bond acceptor is 5, number of hydrogen bond donor is 2, the topological polar surface area is 89.59, log p value is 2.50, it is having 2.78. Okay, approximately it is all the things are similar. The log S value is minus 3.73 and here it is minus 2.70. You can see that the log P means the water solubility is slightly decreased. Okay, so the water solubility is slightly decreased, but it comes under the class of soluble. Okay, so you can see when I substituted the OH group of the moxifloxacin here in the NH2, means I have converted the carboxylic acid to amide, there is decrease in the water solubility. Okay. And you can see it is comes under the moderately soluble class. Okay. It comes under the moderately soluble, but this drug comes under the class of soluble. Okay. So this is the difference. You can find out. So it have the high GI absorption. It doesn't cross the blood-brain barrier. It affects the paraglycoprotein and it have the inhibitory activity of the cytochrome P2C19 enzyme. But in this case, you can see that there is no cytochrome P2C19 inhibitory activity. Means it is inhibiting the enzymes that is responsible for the metabolism. Okay, these are the metabolism enzymes, cytochrome P enzymes. Okay, so we don't want that the enzymes would be inhibited. So if the enzyme is get inhibited, there is reduce in the drug activity takes place. Okay. Then it is also following the Lipinski rule of five, and the synthesis accessibility is also the same as compared to the moxifloxacin. Okay. So in this way, you can just prepare your own setup library. Like you can prepare 5 to 10 setup library, and you can just retrieve all the data in the Excel sheet format, or you just simply screenshot these things. Okay. Now next come to the show the boiled egg. Okay. So what is boiled egg? You can see that the boiled egg is the yellow color represents the blood-brain barrier here, and the blue color represents the paraglycoprotein inhibitor or not. Okay, so as you can see, it is the blue line. That means blue dot is there, means it is inhibiting the para-glycoprotein enzyme. Okay, if not inhibiting, then it will be shown in the red color. But as it is inhibiting the para-glycoprotein, it is shown in blue color. And you can see that these two drugs, these two molecules are not crossing the blood-brain barrier. Okay, if they crosses the blood-brain barrier, they may show some effect in the brain. That may be positive effect or that may be negative effect. But many of the cases, it is found that apart from the CNS drug that crosses the blood-brain barrier, they show the negative effect. Okay, let's take the example of sulfonamides. There is one drug that is called as the sulfadiazine that crosses the blood-brain barrier, and in many of the children, it shows the jaundice. Okay, brain jaundice is there. Okay, so this is the Swiss ADME database. By using this Swiss ADME, you can perform all these things, find out all these things, and this is very much important for your publication figure. And all this information are needed for the uh, research article also. Okay, so now coming back to our slides. Okay, I hope that all of you have understand all these things, the structure-based drug design, ligand-based drug design. Okay, now let's come to the drug repurposing studies. Okay, so now talking about the drug repurposing studies. So basically, what is drug repurposing? You can see here that we have marketed approved drugs. Okay, marketed drugs we have. We have marketed drugs like zidupudin, remdesivir, carbamazepine. Okay, all these three drugs have their different properties. So the zidupudin, it is an antiviral drug. It is an antiviral drug. The remdesivir, it is also an antiviral, and the carbamazepine, it is used in the sedative and hypnotic property. It is used in the sedative and hypnotic. So to hypnotize or to calm down a person who is overexcited, to calm down, the carbamazepine is also given. But what is drug repurposing? Drug repurposing means we have to find out a drug molecule and we just explore another activity of the same drug. Means we can see here the pseudoprotein, which is an antiviral drug, it is repurposed and it is find out that it has some activity to HIV. Means it can have some effect, the positive effect toward the HIV. Means it can inhibit the HIV virus, human immunovirus. Okay. So the next drug, that is the remdesivir. So the remdesivir have the affinity or to ability to cure the COVID-19, not to cure, but to suppress the COVID-19 activity. So as we all know that the COVID-19, it is an CoV-2 virus, CoV-2 virus, and it is an RNA virus. Everyone will know that it is an RNA virus. So in the same case, talking about the carbamazepine, this carbamazepine, it is also effect apart from hypnotic and sedative, it is also used in the treatment of trigeminal neuralgia condition. Okay, so one drug is having two different activities. That is called as the repurposing study. So how can we find out this repurposing study? By performing the in silico analysis. Okay, by the in silico approach, we can predict or we can identify the marketed drug and we can repurpose it differently for the different disease cure. Okay, so you can see here a person is having a symptom A and the drug is given by the doctor. So now this drug is having an effect to cure the symptom B, it may be cured symptom C. Let's take an easy example, the paracetamol. So the paracetamol is nothing but it is a drug to cure the flu. Okay, it can cure the flu, but also it acts as if a person is having a flu, the person is having the headache is there, back pain is there, back head pain is there, then that person is having sneezy nose is there, then watery eye is there. Okay, so paracetamol uh, act as an analgesic means it reduces the pain. Also, it act as an anti-inflammatory. Also, anti-inflammatory. It can cure the symptom B. The symptom A, the main symptom that is the analgesic activity to reduce the pain or to reduce the fever. It also acts to reduce the symptom B associated with the flu. Means it, some it is not completely curing the anti-inflammatory source activity, but however it shows some sort of anti-inflammatory activity also. Okay, let's discuss briefly about this in the next slide. So, what is drug repurposing study? Let me change my ink to red. Okay. So, a drug repurposing study is a research approach in which the existing or already approved drug is investigated for a new disease or therapeutic use. Okay, different from its original indication. So, in drug repurposing study, let's suppose we have the paracetamol 500 mg. We can change that paracetamol 500 mg to paracetamol 250 mg, or we can improve the paracetamol 500 mg to 650 mg. Okay, in this way we can change the amount of the paracetamol also. By changing it may show other activity. Okay, so what are the drugs we can use for the repurpose? So if the drug is known and it has some activity to any disease, we can go for that. And also, if the drug safety is known, we can use that drug. So in this case, we don't need any clinical trial. The development time is much shorter. We don't, here also we are performing the drug discovery, but in a easy way by just repurposing. Means if the some drug is happening a activity of A and we are using for the different disease. That's it. The cost is much lower than the new drug discovery. Okay. So paracetamol is already preparing in the industry. We just prepare the same paracetamol, but with less dose or higher dose. So that will be easy to synthesize. The cost will be low and faster clinical application. Okay, it have the faster clinical application. So these things are done by the pharmacologist. All these things are studied by the pharmacologist. So pharmacologists are there in many medical colleges like AIMS, like all other organization. So there are some recruitment is there for the pharmacologist. So their main focus is on the drug repurposing. They do focused study on the drug repurposing. Okay. So let me give you some of the example of drug repurposing. So you can see here these structures are drawn by using the scheme draw. So first drug here we have that is the amantadine. So this structure is called as the amantadine. So amantadine it is an antiviral drug. It is an antiviral drug. Okay. So primary amine, this NH2 group. Okay, primary. This is the primary amine. This is responsible for its antiviral activity. Okay. But somehow it is found that it is also having some effect in the Parkinson disease. Okay, to treat the Parkinson patient. So, what is Parkinson? You can, in your society, you have seen there are some peoples which are having shivering hand. Their hands shiver. Okay, they are unable to eat properly. because of that shivering. They behave differently. Okay. Then they have slurry speech. They're unable to speak properly. So the amantadine, it is after repurposing, it is found that it is having some sort of affinity to cure the Parkinson. Okay. So this is a drug-induced extrapyramidal symptoms. Okay. So why we repurpose these drugs? So amantadine have the property that it can cross the CNS. As I have mentioned in the case of moxifloxacin, some drugs that may enter into the CNS shows the positive side or that may show the negative side. But in case of amantadine, it shows some of the positive side. Okay, so it in Parkinson's disease, it is useful to treatment of the Parkinson. So the next drug it is very much known that is the aspirin. It is the aspirin. Okay, it is one of the anti-inflammatory drug. Aspirin, analgesic activity is also there. So you can see that this is the salicylic acid. Is there. This portion it is the salicylic acid. Okay, if I place this OH here, then it will be called as the salicylic acid. Okay, so the main use of this drug aspirin is the analgesic. It is having the property of analgesic, reduce the pain, then antipyretic. So antipyretic means to reduce the flu, then the anti-inflammatory to reduce the pain. Okay. Any inflammation is there, we can give aspirin. So when the drug is repurposed, it is found that it is having the anti-inflammatory as well as the anti-proliferative activity. Also, aspirin at the lower dose, that is 250 mg of aspirin, act as a blood thinner. It is act as a blood thinner. So what is blood thinner? So aspirin on 500 mg have the activity of analgesic, antipyretic, and anti-inflammatory. And if the same drug given at the 250 mg, it acts as a blood thinner. Okay. So what is blood thinner? Let me show you here. So this is, let's suppose this is your blood vessel. Okay. The cholesterol is get deposited here. The cholesterol amount is deposited here. So due to this cholesterol deposition, the blood flow let get disturbed. Okay, that may lead to severe heart related problem like heart stroke. So when this drug is given in the lower dose, that is 250 mg, there might be chance that it makes the blood thinner and the blood passes freely. In the emergency condition, another drug is given, that is the nitroglycerin. Many students have known about nitroglycerin. It is given sublingually, that is placed below the tongue. Okay. So aspirin also given in the emergency condition. It acts as a blood thinner. Okay. So the next, that is the, it is one of the other drug. It is an anti-cancer drug. Okay. It is an anti-cancer drug, and it is also having many different some sort of activities apart from anti-cancer. So the cancer are of various types are there. So cancer is having the various types. So it has a broad spectrum of activity in the treatment of cancer. Okay. So it is developed for only treatment of some sort of cancers, but it is find out that it is apart from cancer, it is having other activities like calcium channel modulator. It modulates the calcium channel. The calcium channels are basically, they block the heart signals. They are responsible for the sodium gated ion channels, calcium gated ion channels. These channels are generally used for the circulation or the neurotransmission inside the heart. Okay. So another examples are another two examples. So the first structure we can see here, that is the chlorambucil. This one, the chlorambucil. So the chlorambucil having the main activity of anti-cancer. The main activity is anti-cancer, but after repurpose, it is also used for the antibody. It's used as the antibody drug conjugate. Okay, means it acts as an antibody, and other drug has been attached to it, and it is specific cancer binding activity. It is okay. It is targeted cancer delivery system. Okay, so it is also having a broad spectrum of the anti-cancer activity. It is DNA alkylating activity is there. And the final one, it is the sulfonamide moiety. You can see sulfonamide. This one is a sulfonamide moiety. So it is the drug that is having the original use. It is an sulfonamide antibacterial activity is there. This drug is having the antibacterial property. So after repurposing, it is also having the carbonic anhydrase activity means diuretics. It is having the diuretic activity. Also, it is having the anti-inflammatory activity. Okay. So these are some sort of examples of drugs for the drug repurposing study. So amantadine, the first one is the amantadine. This one is the amantadine. This one is the aspirin. Then this one is another drug. We have here another drug. This is the chlorambucil is there, and this is the sulfonamide derivatives. Okay. I hope you guys have understand all these things. So the drug repurposing is having three types. So network based, first one is the structure based drug design or the surface structure based drug repurposing, ligand based drug repurposing, and the network based drug repurposing. Let's go through it. Okay. So what is the workflow? So we are going to study about this in the upcoming sessions also. So the workflow in the structure-based drug design, so selection of the disease relevant target. So first we have to select the disease relevant target by using the PDB. Then we have to prepare that protein or to prepare that receptor. Then we have to select the FDA approved drug library. Then we perform the docking. We validate the score and rank the drugs. Then we analyze the interaction by using Discovery Studio. Then we validate using the redocking process and calculate the RMST and RMSF value. Okay, so it is very easy process. We have to select the target first. Then we have to select the drug molecules. We have to prepare a list of drug molecules. Then we perform the docking. Then we score them. We rank them. Then we analyze all these things and go for the simulation studies. Okay. Then the ligand-based drug repurposing. It is the drugs with similar chemical feature often show similar biological activity even on new targets. Okay. So the ligand-based drug design is nothing but the broad activity. While I am saying about the broad spectrum activity, so the ligand-based drug repurposing is following the broad spectrum activity. Okay. As I have shown you the pyrimidine moiety. That pyrimidine moiety is having various activities, like it is having antimicrobial, it is having antioxidant. So if I consider the pyrimidine moiety, this moiety, and I started to repurpose this drug, so it can have various numerous number of activity. So where to find out these things? So we can perform some techniques like chemical similarity searching, pharmacophore modeling, QSR. It is very much similar to the structure-based drug design and ligand-based drug design. So what are the database we can use? So we can use the ChemBBL, PubChem, drug bank. So these three databases we can use to find out the ligand molecule for our drug repurposing. Okay. So in this session we have learned about the PubChem, and in the upcoming session we are going to discuss about ChemBL drug bank, and we'll also study about the preparation of the ligand molecule and the preparation of the receptor molecules. So how we represent our ligand molecule in the LBDD. In the LBDD, ligand-based drug design, or in the drug repurposing. So these are the formats in which we can save our ligand molecule after preparing. So the first one, it is a photographic format. You can see this is a JPEG format which I have given here also. Then the PDB QT format is there, PDB protein data bank format is there, Mol2 files are there, FTF format is there, and the simplest smiles representation is there. So these are some sort of formats in which our ligand molecule can be stored. Okay. So thank you everyone for joining this session. And in the upcoming session, we are going to learn about all these things. Let me write it down here. We'll learn about the databases. We protein, retrieve the proteins using the RCSB-PDB. Then, thirdly, we'll prepare the protein using the tools that are used for the preparation of the protein. Then we retrieve the ligand molecule also, and we prepare the ligand molecule. The energy minimization is there, force field is there. All these things we'll learn in the next session, because this is a very theoretical session and many concepts you have to learn. So that's why I am not stretching this sessions to like 2 hour session. It will make you impatient and there will be no advantages for you also. Okay. So thank you so much for joining the session, and all the doubts you can write in the comment section, or you can drop a message in the group. I will answer all your doubts. Please watch this video till the end. Okay, so that you can have a brief idea on all these things. So thank you so much for joining, and keep supporting and keep learning. Happy learning, all of you. Thank you so much.